Okay, today we are going to work on a virtual lab on susceptibility testing. We're going to really discuss the susceptibility testing that is done manually in a laboratory. There's a couple of rapid tests that we will do, as well as set up and interpret susceptibility testing that is done manually. Really, in most clinical labs, most susceptibility testing is done with using commercial systems. But today we're really just going to look at some of the manual setup and interpretation of susceptibility testing that we can do outside of commercial systems. So we're going to start with setting up disc diffusion susceptibilities, okay, otherwise known as Kirby-Bauer test. So this process is we use filter paper discs that have already been impregnated with antibiotics, and they are placed on solid media such as the Mueller-Hinton agar. And what we're going to do is we are going to streak these plates for lawn growth. So lawn growth is rather than individually isolated colonies that we really like to work with when we are identifying organisms. But lawn growth is when you get a nice smooth set of growth that is not individual colonies. Here's an example, and we'll come back to this when we interpret it, but here's an example of lawn growth. You can see there's just one. You can't see individual colonies. It's just a smooth suspension growth over the whole plate, okay? And that's what we prefer when we are actually interpreting growth for Disc Diffusion and Etest as well. So what we do is we will streak out these plates, wanting a lawn growth of the organism to be tested. And then after that organism is incubated, that suspension is incubated overnight, then we will come back to that plate and we will observe and look for a zone of inhibition around the antibiotic disc, and we will measure that. The size of the zone of inhibition is actually inversely proportional to the minimum inhibitory concentration of the organism. Okay. Now for these discs, they are impregnated with a set concentration of antibiotic. Okay, so it's one set concentration, and then we are looking to see whether at that set concentration if the organism is susceptible or not. Okay. So we're going to start. We're going to use sterile saline, and I'm going to start with an organism that is a known, this is a known Staph aureus, okay? And I'm going to inoculate a tube of sterile saline, okay? Now typically we would want to vortex this. I don't have my vortexer out with me right now, and I would actually measure this to make sure that I have a 0.5 McFarland standard inoculation, okay? I'll pull that out for you a little bit later and show you what those standards look like, okay? So I'm going to label my plate Staph aureus. Now I'm going to take my inoculation and I'm going to streak. You notice I'm going to kind of rim that swab a little bit so that I don't actually have too heavy of a suspension. Now I'm going to inoculate this plate by streaking in for a lawn growth. Do you see that I'm streaking the entire plate? I'm going to turn it 45 degrees, streaking it all the way across the plate again. And one more time, 45 degrees, streaking it all the way across the plate. I'm going to rim the whole plate like that. And if you can see the lines of the streaking on this plate, you're going to notice that virtually the entire plate is covered by streaking. Okay. All right. If you happen to streak for isolation, you really will get an incorrect result, because you will now have a you will have gaps in your media. Okay. So these are the discs. So the disc I'm using today for this versus the Staph aureus, this is amoxicillin clavulanic acid. Okay, it's a combination drug. Okay, I have sterile forceps here, and I'm going to carefully place that on my plate, and I'm going to kind of tap it down so that that it's not going to fall off when I turn it upside down, because I'm going to incubate it this way. Okay, I'm going to put that to the side. That will get incubated overnight, and we will actually set that, we will interpret that tomorrow. Okay. So I'm going to pretend, now this is not a, this is actually not the same disc, but I do have an example of a plate that has been inoculated. It was set up for a disc, and this actually is a gram-negative bacilli called Stenotrophomonas, and this is a minocycline disc. Okay. Now, in order to read the zone size, we have a really handy tool called a caliper. Okay, you can just use a ruler, that works as well. But these are very, very precise. You can see it's kind of got, I'm able to adjust it. The large kind of rulers part here, these are, this is, each little dot represents 10 millimeters. And then this dial is set up for 1 millimeter each, okay? So I want to read the diameter of the zone of inhibition, okay? So you want to use reflected light, okay? And you want to be able to measure the diameter of the zone of inhibition, okay? So if I set my caliper on either side of that zone, it is 20, because I can see it's 10, 20, 25 millimeters, okay? I'm going to write that, on my plate, because we're going to come back to that. We're going to look up the interpretation in the CLSI document that is our standards for susceptibility testing. And so we are going to use that document in order to interpret our susceptibility testing for the disc diffusion. Okay. Now I'm going to set up, the next test I'm going to set up is my E-test. Now E-test use E-strips, and these are non-porous plastic antibiotic strips. Now these are different. They're different than the Kirby Bauer discs, because remember, those were set at a set concentration on each disc. The E-strip is calibrated with MIC values that cover a wide concentration range. So a predefined antibiotic gradient is immobilized on the surface of the strip. We take that strip and we place it on the Mueller-Hinton auger, which has been streaked for lawn growth, just exactly how we did the disc diffusion. Okay. When we transfer the strip to the auger, the gradient remains stable as the organism grows. So that means that a varying amount of concentration of the antibiotic is being released into the auger. So the MIC, the minimum inhibitory concentration, is actually determined by an E-test. It's red where growth intercepts the strip, okay? So this time, so we're going to set up another Mueller-Hinton plate. Okay, this time we're going to use an E. coli. Going to make our suspension and our sterile saline. Again, we're going to pretend that we have our instrument called the density check that allows us to measure precisely. Right now I'm just shaking it. Typically we'd vortex it and we would make sure that that concentration is exactly between 0.5 and 0.63 is the concentration that we want to use. Okay. And if you look, you can see there's a density you can kind of see that there is a solution in there. Compare it to my negative tube. You can see the one on the left is a little bit turbid, okay? All right, so again, I'm going to take that, dip it in, that swab in. I'm going to make sure I get any excess liquid off. I'm going to get my Mueller-Hinton plate, and again, I'm going to streak it for lawn growth. I want the entire plate to be covered, a nice even growth. So there's no gaps or bumps or areas where you can see gaps. Those gaps can give you incorrect looking like there might be some area of inhibition when in fact it's just an area that the organism wasn't actually growing. Okay. I'm going to label this as E. coli. Okay. Okay. So this time I'm actually using something, the Etest. Now, just so you know, I'm using actually expired discs and Etest. So these are not necessarily, the ones that I'm setting up are not necessarily the discs or the strips that I would use on this particular organism, because like I said, I'm using what I have access to, so I'm not actually wasting material. So now what I'm going to do, I'm going to kind of break this. Okay, you see this strip kind of comes up. Again, I don't want to touch that strip because that strip has gradient of antibiotics on it. So using a sterile forceps again, I'm going to gently lay this down, and I'm going to carefully make sure that it's going to, that I'm going to go from the least concentrated to the most concentrated, and I'm going to touch it just to make sure that it's not going to come off. Now here you can see the top has the highest concentration. Now this is daptomycin. So actually daptomycin really is not active on E. coli. I'm just using this as an example for the setup of it. The highest concentration of the daptomycin is 256 micrograms per milliliter. And the lowest concentration is at the bottom, 0.016 micrograms per mil. So what you will find is that when you will be looking for a zone of inhibition, and it's going to look like an ellipse. So the larger zone will be at the top, at the most concentrated section of the strip, and the smaller area of inhibition will be at the bottom where it's less concentrated. Okay. All right. So once again, this would be incubated overnight. All right. So now I'm going to actually show you. This is a daptomycin plate. Okay, this is a large plate. It's a little bit hard to read. You really need to read these so you can see the zone. I know it's hard to see because this organism does the lawn growth is actually very, very thin, very small. So let's see if I can point out to you. See if you can see. I'm going to touch it from the bottom of the plate. Here is your inhibition. that ellipse, okay? So where it's actually intersecting with, on either side, with that strip is right about at 2, okay? That 2 is represents micrograms per mil. So 2 micrograms per mil would be what the reading of the Etest would be, okay? Now again, what's interesting about the difference between the Disc Diffusion and the Etest is that since the Etest actually has a gradient of antibiotics, you are actually able to read an MIC. Okay, you are actually, because you have a range of the concentration of antibiotics, you can see exactly where that concentration is going to inhibit the growth of the organism. Whereas the Etest, whereas the Disc Diffusion will only give you an ability to determine whether it is sensitive, resistant, or intermediate. The Disc Diffusion or Kirby-Bauer does not give you an MIC, okay? Only whether it's resistant or susceptible, okay? Here's a couple other Etests, okay? Different organisms, different antibiotics, okay? This LE, that represents levofloxacin. See if I can get that out of the way. Maybe that might help you read that a little bit more, okay? Okay. I'm reading the zone of inhibition to be right at about 1.5, okay? That's where it is. That is where it is being completely inhibited. Now, talked a little bit about, in class, we've talked a little bit about how different antibiotics, they can either be bactericidal, which means they completely kill the organism, or bacteriostatic. Antibiotics that are bacteriostatic sometimes will have to be read, the zone has to be read at maybe 80% inhibition rather than 100%. So for instance, I don't know if you can see, this is kind of might be a little bit hard to read. The TS is trimethoprim sulfa. This is a bacterial static organism, and so its zone of its inhibition is read at about 80% inhibited rather than 100% inhibited. So in this case, we're going to read at about, well, because actually it's hard to tell. It's actually, there's a little bit of growth almost over the entire kind of that big ellipse. There's actually a little bit of growth all the way up to the highest concentration, which is 32 micrograms per mil. But right at about 1.0, there is actually some inhibition. So it's kind of hard to see on this one, but we would read this at that 1.0, which is where you've got about 80% inhibition, okay? So that's how you interpret, or that's how you read and determine the actual reading for these organisms, okay? So now I'm going to pull the document for the CLSI so we can actually determine what those numbers mean. Okay, so I took out just one, because this document is so huge, I actually took out a page, the page that is going to help us determine the daptomycin result. If you remember, the daptomycin result was 2 micrograms per mil. That's where the zone was, the ellipse, and the organism was Enterococcus, okay? So we are going to look at daptomycin, and we had a daptomycin that was an Enterococcus faecalis, okay? So now I'm looking at the interpretive category for the MIC breakpoint, because once again, the Etest is an MIC. It says if it's less than or equal to 2, then it's susceptible. If it's 4, it's intermediate. And if it's greater than or equal to 8, it's resistant. Our Etest for that daptomycin was 2 micrograms per mil. So I can see the 2 micrograms per mil is susceptible. So in this case, our daptomycin would then be interpreted as susceptible versus the Enterococcus faecalis. And all the way through that document are all different kinds of organisms, all antibiotics that are appropriate to be appropriately used to treat that organism, and that's how we determine whether or not it is acceptable, or the physicians will contact us and ask whether or not the organisms are susceptible against those antibiotics, and so we use that standard as how we help determine that information. Okay, the next susceptibility test that we're going to do is called the beta-lactamase, and we're going to use what's called the cephinase disc. So the disc is, once again, this is another disc that has cephinase, which the cephinase. I'm sorry. Let me back up a minute. So we are. Sorry. Stop this now.